A very good afternoon to everyone. Welcome in the class. The topic of today's class is southern blotting. We are going to talk about various blotting techniques in the further classes. The topic of today's class is southern blotting. So just give me a second. Yes. The topic of today's class is southern blotting. And as we say in the starting, let's crack CSIR UGC NET together. Let's crack GATE 2021 together. GATE 2021 together. We're watching this video under the platform of Unacademy, which is one of the India's largest platform for the preparation of CSIR NET examination and various other examinations. I'm your educator Farida Johar Bandukwala. I have a teaching experience of 10 years. I have qualified MP SLET examination in year 2018. If you're opting for Unacademy, what all things you are going to get? You are going to get daily life classes so that you can be always in that momentum of studies, and live classes means you can always ask your doubts and queries directly with the faculty member in the class. The whole syllabus is quite structured. That means that the whole syllabus is very lengthy, so in a very precise manner and within the limits it will be covered. Live tests and quizzes are taken. That means that you can evaluate yourself while the class is going on and while you are preparing, and unlimited access towards all the videos and study material which is particularly present in that category. Free live classes are the classes which are conducted on two platform, that is YouTube, and it is conducted on special platform. So for watching the free classes on YouTube, you just need to download the Unacademy learning app. When you will download the app, you will be asked to make your account. After making account, you will have to fill the referral code of an educator. You can fill my referral code, that is FP01, and after that you will get the access to for watching all the special classes on the Unacademy platform. That is the Unacademy learning app. Then there is this plus subscription, and this plus subscription is a paid subscription. You need to pay a certain amount, but the amount is very pocket friendly, and recently a major discount is going on, so you can avail this discount and take the plus subscription. For 12 months it is going to cost you 11,500 rupees, and for 24 months it is going to cost you 16,800 rupees. You can get an additional 10% discount by using my referral code FB01. Do not forget to use my referral code, that is FP01, and the 12 monthly subscription is going to cost you only 10,395, and 24 monthly subscription is going to cost you 15,120 rupees. So that was the small introduction about Unacademy. The topic of today is southern blotting. We'll be talking about southern blotting. Southern blotting. Before beginning with the actual topic, that is southern blotting, let us have the meaning of blotting. That if we are saying blotting, then what does this mean? Blotting. So a blot is nothing but it's a biomolecular or molecular biology and genetic word, and it refers to a method in which DNA, RNA are transferred on a carrier. DNA, RNA, and proteins that are transferred on a carrier. So a in molecular biology, blot in molecular biology and genetics is a method of of transferring proteins, DNA, and RNA on a carrier. Right? We'll be talking about carrier and everything, but just first of all try to understand the meaning of blotting. Then the term blotting, it also refers to the transfer of biological sample from basically a gel to a membrane, and their subsequent detection on the surface of the membrane is also referred as blotting. So what does this blotting means? Transfer of biological sample biological sample from gel to a membrane, and further its detection is done. Further its detection done. This is blotting. So technique used for transferring DNA, RNA and proteins onto a carrier so that they can be separated, and often follows the use of gel electrophoresis, because if we are having DNA sample or protein sample, we need them to be in distinguished samples or distinguished places. So for separating out DNA, RNA and proteins on separated out form, usually gel electrophoresis is performed. So technique for separating out or separation, technique for separation of proteins, DNA and RNA is gel electrophoresis. Gel electrophoresis. Fine. So there are various types of blotting. So blotting techniques are itself of three types of blotting. So in today's class we are talking about Southern. So Southern blot is for detection of DNA, detects DNA. Then northern blot is for detecting the RNA, detect RNA. Then western blot is for detection of protein, detect protein. So these are the three types of blotting techniques. Now the topic of today's class is Southern blotting. So we'll be talking about Southern blotting. Southern blotting. Right. So Southern blotting or Southern blot is a method which is used in molecular biology for detection of specific DNA sequence in the DNA sample. So method for detection of specific DNA sequence, specific DNA sequence in DNA samples. DNA samples. This is the first thing. Second, Southern blotting is actually a combination of two processes, that is combines the transfer of electrophoresis separated DNA fragments onto a filter membrane, and then after transferring them on the filter membrane, their subsequent hybridization with the probe for the detection of a specific sequence. So there are totally three things which are involved in Southern blotting. First is gel electrophoresis. After gel electrophoresis, transfer onto filter membrane. After this, detection by probe. Detection by probe hybridization. So basically, when we are talking about Southern blotting, we are talking about how many things? First of all, gel electrophoresis would be performed. Then it would be transferred onto the filter membrane. And after that, when it is transferred on the filter membrane, its detection would be done by probe hybridization. We'll be talking about each and every step in detail. So this is just the introductory part. So listen to this and we'll be moving in the detail thing also. So the name of the method, that is Southern blotting, is given after its inventor. So he was a British biologist. Southern blotting is given by a British biologist, and his name was, obviously somewhere in the name Southern will come. So he was Edwin Meller Southern. Edwin Meller Southern. This is the name. After that, we'll be talking about the principle of the process. So let us write principle. So the key to this method is basically hybridization. The most important thing in this process is hybridization. Key to this method is hybridization. Now, what is hybridization? So it's a process of forming a double stranded DNA between single stranded DNA probe and single stranded target DNA whose sequence is unknown and whose sequence we are actually trying to detect with the help of probe. So hybridization means onto a filter membrane, certain DNA would be fixed and we would know certain sequence probes and we'll be adding them on this membrane, and if they join somewhere, it means sequence complementary to that probe is present on the paper or the filter membrane. So that is what hybridization means. So process of forming a double stranded DNA molecule between single stranded DNA probe and single stranded target DNA. Right? So there are two important features of hybridization. Let us talk about that. There are two important features of hybridization. So the reactions are specific to the probe, or the things which are specific to the probe will only let the probe bind to them. And the probe can find one molecule of target in a mixture of millions of related but non-complementary molecules. So two things are there, that probe is not going to bind anywhere, and in millions of reaction mixture, probe is going to bind to just one. These two things are very, very important when we are talking about probe. So specificity of probe, specificity of probe to complementary sequence, and another is probe can find one molecule of target in a mixture of millions but related non-complementary molecules, of target in millions of copies. So these are the two important features, and this is what the principle of Southern blotting says. Now we'll be talking about the steps which are involved in Southern blotting. Steps. The first step is extract and purify DNA from cells. Extract and purify DNA from cells. Second is DNA is restricted with enzymes. DNA is restricted with enzymes. Then separated by electrophoresis. They are separated by electrophoresis. Then fourth is denaturation of DNA. Then transferring onto nitrocellulose paper or filter membrane. transfer to nitrocellulose paper. Then add the labeled probe for hybridization. Addition of labeled probe for hybridization. Then washing off of the unbound probe. Washing off unbound probe. And lastly, auto radiograph. These are the 8 steps which are involved in this whole process, right? Now there is a apparatus which is actually used for this process. So we'll be seeing first the apparatus in which most importantly the transfer occurs. So there will be this rubber plate. In this rubber plate there will be filling of transfer buffer. There will be filling of transfer buffer. Then there will be platform. On this platform there will be a wrap. Onto this wrap there will be a Whatman paper. This is Whatman paper. On this Whatman paper there will be placement of a gel. This is the gel which will be kept over it. On this gel there will be a stack of Whatman papers. Stack of Whatman papers. So we'll be writing Whatman paper stack. Whatman paper stack. And on this there will be placement of paper towels. Paper towel stack. Paper towel stack. And finally on the top there will be placement of certain kind of weight so that the things are under a pressure. So this is weight. This is the whole apparatus of the process. Like there will be different apparatus for hybridization, different apparatus for electrophoresis. But when we are actually transferring the DNA from our gel to nitrocellulose paper, this apparatus is used. So you will be writing here for transfer of DNA onto nitrocellulose paper. This thing is done. So next thing that we are going to see is now stepwise how the southern blotting is performed. So first is extract and purify DNA from cells. Extract and purify DNA from cells. The first thing is isolate the DNA which is in question from the rest of the cellular material in the nucleus. Isolate DNA from cells. Then incubate the whole specimen with detergent for promotion or promoting the cell lysis. So isolating the DNA from cell, how will it be done? By incubating specimen with detergent to promote cell lysis. Promote cell lysis. Then lysis free freeze the cellular proteins and DNA. Lysis freeze cellular proteins and DNA. Then proteins are enzymatically degraded by proteases. Proteases for protein degradation are used. Protein degradation are used. Then organic or non-inorganic extraction removes the proteins and DNA is purified from the solution by alcohol precipitation. DNA is purified by alcohol precipitation. So the DNA fibers which are visible, they are removed and they are finally suspended in the buffer. So this is the first step, that is the extraction and purification of DNA. Second is DNA is cut out with the restriction enzyme. DNA is cut with restriction enzymes. So what will be done? The DNA would be incubated with the enzymes. These are the enzymes. DNA would be incubated with enzymes, and after incubation we will be getting small fragments of DNA. Small fragments of DNA. So this is digestion with restriction enzymes. Digestion with restriction enzymes. This is the second step. Then the third step is separation by electrophoresis. Separation by electrophoresis. So the complex mixture of fragments, they are subjected to gel electrophoresis for separation of fragments according to the size. So separation by electrophoresis on basis of size occurs, right? So we have seen the process of gel electrophoresis, isoelectric focusing, 2D gel electrophoresis. So we know that how the separation of electrophoresis is going to occur. So it is actually based on the size when we are simply talking about electrophoresis. Then next is denaturation of DNA. Denature DNA. So the restriction fragments present in the gel are denatured with the alkali. So the bands which are present on the gel, they are denatured with the alkali DNA strands in bands on gel are denatured with alkali. This causes the double strands to become single stranded. This causes double strands to become single strands. Then DNA is neutralized with NaCl to prevent the rehybridization before adding the probe. DNA is neutralized, DNA is neutralized with NaCl to prevent rehybridization. So this is the process. So why is it neutralized? So that it do not rehybridize before the addition of probe. Before adding probe. This is the process of denaturation. After this denaturation has been done, the next step is transfer onto the nitrocellulose paper. Transfer onto nitrocellulose paper. So transfer the DNA from the gel to a solid support, that is called as blotting. Transfer of DNA from gel to a solid support is blotting. Is blotting. So the blot is made permanent when it is already transferred. Then it is made permanent by baking it or drying it at approximately 80 degrees Celsius. Made permanent by drying, which is also called as baking, drying at 80 degrees Celsius and exposing to UV irradiations. Exposing to UV. This is the fifth step. That is the transfer to nitrocellulose paper. Then the sixth step is addition of probe for hybridization. Addition of labeled probe for hybridization. So the filter is incubated under the hybridization condition with a specific radio labeled DNA probe. So for example, this is the chamber and here is the nitrocellulose paper. It will be flooded with the probes. So probes would be there overall in this mixture. So they will be led to hybridize, and when it will hybridize, then there will be removal of some unnecessary unbound probes. So let us write the filter is incubated. The filter is incubated under hybridization conditions with the specific radio labeled DNA probe. The radio labeled DNA probe. So the probe they hybridize to the complementary DNA and unbound probes they are washed out. So next step or here we'll write probe probe hybridizes to the complementary DNA to the complementary DNA. After this, the next step is wash of unbound probe. Wash of unbound probe. So blot is incubated with wash buffers which are containing the NaCl and detergents for washing away the excess probe and reduce the background. So wash buffers containing NaCl and detergent to remove excess of the probe is done. After this, lastly, the step that is performed is autoradiograph. Autoradiograph. So if the probe is radioactive, the particles are going to emit when they are exposed to the X-ray film. So there will be dark spots on the film wherever the probe is bound. So autoradiograph is performed if it is radioactive in nature. So this is how the Southern blotting is performed. So we have seen the process of Southern blotting. Now we will be talking about its applications. So they are the Southern blotting technique is used to identify specific DNA in a DNA sample. Identify specific DNA in a DNA sample. Then to isolate the desired DNA for construction of rDNA. Isolate desired DNA for rDNA. Then it is also used for identification of various mutations, deletions and gene rearrangements. Identify mutations, deletions and gene rearrangements. After that, it is used in prognosis of cancer and in prenatal diagnosis of genetic diseases. Used in prognosis of cancer and various other prenatal diagnoses as well. Then it is used in RFLP. Used in RFLP. Then it is used in diagnosis of HIV and various infectious diseases. Diagnosis of HIV. Then it can also be used in DNA fingerprinting. And DNA fingerprinting we have done in yesterday's class. So a whole lot lecture is available on DNA fingerprinting. I am not going in detail that it is used for paternity and maternity testing, criminal identification, personal identification and lot many things. So we are not going in that detail because we have done it in yesterday. I'll be talking about the advantages and then disadvantages of the northern blotting. Disadvantages. What are the advantages? That it is an effective way to detect specific DNA sequence in large complex sample of DNA. Effective to detect a specific DNA sequence in a large complex sample of DNA. Complex sample of DNA. Then it can also be used to quantify the amount of the present DNA. Quantify DNA. Quantify DNA. And this process of Southern blotting is actually cheaper than DNA sequencing. Cheaper than DNA sequencing. Then what are the disadvantages? That it is complex as compared to other techniques because it is composed of various steps. Like just now we have seen there are total 8 steps by which Southern blotting is completed. So it is complex. Then, sorry, it is labor intensive. Then it is time consuming and cumbersome. time consuming and cumbersome. So like this, we have completed this process of southern blotting in detail, and I hope you people have understood well. And as we say in the starting and end, let's crack CSIR UGC NET together, and let's crack GATE 2021 together. If you haven't subscribed to our channel till now, please subscribe to our channel, that is Let's Crack CSIR UGC NET, and hit the bell icon for getting all the notifications. If you really like my video, please give it a thumbs up. It really motivates the educators like us. And recently a discount is going on in the Plus subscription, so do not forget to use my referral code FB01 for getting a 10% discount. The Plus subscription can really help you qualify the examination, because each and every uh, topic which is described in your syllabus is explained in the Plus subscription. So go for the Plus subscription, and you can attend the special classes which is conducted on YouTube Learning app. These special classes are completely free. You can use my referral code FB01 for attending the special free classes as well. So I hope you people have understood the things well. Thank you to one and all for your patient listening in the class, and hope to see you tomorrow morning with another interesting topic of CSIR syllabus in the morning at 10:30, and at 5 p.m. At 3 p.m. we'll be doing an interesting topic of GATE syllabus. So hope to see you at 10:30 in the morning and 3 p.m. in the noon. So till then, be happy and keep on studying.